So it's a total honor and privilege to be here with you today to give the TRACE opening remarks. I think we've come a really, really long way in this program in the past 11 or 12 years. And so I'm excited to talk about where we've gone and where we're going. Okay. So I am Passley Hargrove-Grimes. I'm a program officer at the National Center for Advancing Translational Sciences and under the directorship of Danilo Tagle, who many of you know. And I want to just thank the organizers of the Critical Path Institute for the opportunity to talk to you today about how we are funding translational centers for microphysiological systems and how our ultimate goal is to really qualify some of these systems as bona fide drug development tools. So before I get started on what our translational centers are about, I want to talk about where we've been and really lay the foundation for where we're going. So we started really with low-hanging fruit about a little over 10 years ago with safety pharmacology studies, really bringing together NIH, DARPA, and the FDA to support the development and integration of these multi-organ systems and the generation of renewable human cell resources for predictive safety assessments. We pushed for human relevant models, particularly for hepatic, neuronal, renal, cardiovascular, GI, and immune toxicities. And the goal really was to assess toxicity where no physiological and pharmacologically relevant models are available. 5 years later, all of our initiatives are 5 years really. So 5 years later, we moved into disease modeling and efficacy testing, where we really hope to capture the pathophysiology, mutation spectrum, and phenotypic diversity of human diseases and conditions. We responded to national health emergencies, which was first and foremost the opioid crisis through the HEAL initiative, and then we went on to study SARS-CoV-2 on CHIP. We've also studied accelerated aging through our CHIPS in Space program, and we just put out a new call for our tissue chips in space 2.0. So we'll have kind of a continuation of that program. And we've also modeled more complex diseases on chips, such as Alzheimer's related dementias, as well as type 2 diabetes in collaboration with NIDDK and NINDS. And then a few years ago, we moved into the precision medicine space, asking ourselves if tissue chips could really be used to inform clinical trial design, planning, and execution, if they could help us establish patient recruitment criteria, stratify patients based on response or lack of response to a candidate therapeutic, develop clinically relevant surrogate biomarkers that represented population and patient diversity for each disease of interest. We've also worked to build confidence in the technology through creation of independent tissue chip testing centers that could onboard our developers' technologies and test them for robustness, reproducibility, reliability, and relevance. And we've also stood up an MPS database where we've hoped to aggregate and manage MPS data, as well as integrate MPS data with human and animal exposure data and to enable the evaluation of MPS results with reference and clinical data. And those tissue chip testing centers, as well as our MPS database, are now fully independent, and we're very excited about that. We've also sought to build community. So we stood up funding for the first 3 years of the MPS World Summit, which Jay just mentioned, and we do really hope that you'll join us. I'm not on the committee, I'm just an avid proponent. And also we've stood up the International MPS Society, both of which are now they're fully funded on their own and will be remaining viable. I would be lost without saying that we have partnered extensively with other institutes at NIH. I think at this point, it's over 20 different ICs. We've also worked hand in hand with the FDA, BARDA, EPA, NASA, and the VA, just to name a few, kind of since the inception of the program, as well as the IQ MPS affiliate, which spoke earlier in today's talks. So now our goal is to really push these microphysiological systems into the regulatory space. The purpose here was to really establish centers that support research that will accelerate the translational use of MPS in drug development through regulatory acceptance and adoption for industry use by establishing MPS that are fit for purpose for industry needs and have highly specific defined contexts of use that will be developed with consideration of applicable expectation to achieve regulatory approval. So what we've tasked these 4 groups with doing is essentially to take 4 to 5 validated MPS with specific contexts of use. And we've talked a lot about validation today, but these models have been published on, and a lot of them have been reviewed through the tissue chip testing centers as well. for various purposes. So for safety farm, for disease modeling and efficacy testing, as well as for precision medicine. And then to work kind of hand in hand with the U.S. Food and Drug Administration, in addition to the Critical Path Institute, to qualify these along a pipeline. Now, whether that's through ISTAND or through biomarker qualification, or through a similar mechanism. And so hopefully, once these are qualified, making them publicly and commercially available to fill unmet needs in drug development. So just in terms of the overall governance of Trace MPS that I wanted to point out, we are, NCATS is working with the National Institute on Aging, as well as the Office of Research on Women's Health, to provide the overall governance structure. We have monthly calls with our awardees, our FDA subject matter experts, as well as our program staff. We meet quarterly and have pre-LOI calls with our awardees, with FDA regulators, as well as CPATH representatives. And our PIs have recently organized organically by themselves their own intercenter working group, which we're very excited about. And so I just want to list, this is a list of the 4 major projects that we're funding. We have Jonathan Himmelfarb's team from Mount Sinai and University of Washington, Seattle, standing up a translational center for kidney microphysiological systems to improve drug safety and efficacy. Jim McGrath's team at University of Rochester is standing up a translational center for barrier MPS. Ron Menon and Yvonne Rusin are standing up a center from UTMB on a translational center for microphysiological systems-based drug development tools for pregnancy and women's health. And then Lance Taylor's team at University of Pittsburgh Drug Discovery Institute is working to qualify patient-derived biomimetic liver MPS as drug discovery tools for drug metabolism, toxicity, efficacy testing, and clinical trial cohort selection. So you'll hear from them in just a moment. And with that, I just want to say thank you so much to everyone that I work with, particularly Danilo Tagle, who could not be here today, my fellow program officer, Dimitri Krepke, and our program analyst, Chris Sundrick. I also want to thank Tiziana Cogliotti from NIA, as well as Dave Thomas from ORWH, as well as all the individuals listed here who've helped us from the U.S. Food and Drug Administration, as well as the Critical Path Institute. And with that, thank you so much, and we'll have the awardees. Thanks.